So today I wanted to talk to you guys about the RRIDs and cell lines, and what we're calling here naughty cell lines. My name is Dr. Anita Bandrowski. I run the RRID initiative, and I also run SciCrunch Inc. So I have both an affiliation with a company and UCSD. My slides are not working. There we go. Okay, so what are RRIDs? I think you've heard about these before, but these are persistent unique identifiers for the things that scientists actually are using. And so these things typically go into the methods section. They were created jointly by the National Institutes of Health, a group of journal editors that came together at the NIH and several scientific meetings, as well as UCSD and several other researchers. And they function in support of the NIH guidelines for rigor and transparency. RRIDs are funded and maintained jointly by the Neuroscience Information Framework, dkNET, and several contracts from the Office of the Director, as well as the Helmsley Charitable Trust. Okay. Now, the issue of contaminated and misidentified cell lines being in use in many, many studies has been a real problem for a group of researchers in the cell line space. Christopher Korch here, pictured in his 2015 Science article, is really one of the key persons who is really fighting against this, trying to both raise awareness and also has been trying to estimate how many papers are actually affected by cell line problems. Cell line problems really break down into two broad categories. There are contaminated cell lines. So at the stock center, some cell lines become contaminated with other cell lines, which can obviously be very, very bad. If you think you're buying one cell line and you're actually buying a different cell line. And then the second kind of problem is misidentification. And misidentification really comes down to what was first supposed about a particular cell line, but now we know that the cell line is actually something entirely different. So there's an organization, so the problems around cell lines had spawned an organization called Icy Lag. This is the International Cell Line Authentication Committee, which is composed of lots of researchers as well as heads of various stock centers, cell line centers, and they have put out a lot of guidelines in terms of how do you authenticate a cell line, how do you make sure that the cell lines that you're providing to the research community are actually good and authentic. Also, they have actually put out a list of cell lines that are, problematic or misidentified. And these cell lines are on their website. And unfortunately, it is our understanding that, in fact, not all of the researchers out there are aware that Icy Lag exists, and that the list of problematic cell lines can be accessed. So one question to ask, and we don't have a good answer for this out of this community, is how many papers really use cell lines at all, and how many of those papers actually use problematic cell lines. There have been many estimates throughout the years, but those estimates are just that. So what we employed here is a tool called SCIScore, and we trained SCIScore using about 1,200 examples of cell lines inside of sentences from the method sections. Now, these all actually did come from open access papers. SCIScore was then trained using a relatively standard method in text mining, which is the 90-10 split. where you essentially take 90% of the data that you have. So out of those 1,200 examples, you take 90% of those, and you train the algorithm using that system, and then take the last 10%, randomly assigned, of course, of the examples, and then you do your testing on that last 10%. So there were about a little over 100 cell line examples which we tested. And after this training, which we repeated 10 different times, after this training, SCIScore recognized about 70% of the cell lines in any one sentence. And the precision rate, which we set, we were interested in making sure was very high, was 95%. So this is a relatively good text mining algorithm, and this has now also been incorporated into SCIScore, the tool. Okay, so what does recognition actually look like? So here I've got two sentences, sentence 1 and sentence 2. These are both from published papers. The PubMed ID is right there, so you can always find this. This is sentence number 353 in the methods section, where these authors said for luciferase activity assays HeLa or HCN-A94 cells were grown in 24 well plates and transformed with this particular plasmid, et cetera, et cetera. So the reason I brought up these two examples is because I wanted to show what are kind of correct annotations that are being made and what are false positives and false negatives. So this plasmid was actually recognized as a cell line by SCIScore, but the HeLa cell and the HCN-A94 were actually correctly annotated. So in this particular sentence, SCIScore found these three entities. One of these was a false positive, and these false positives are actually very rare. However, they do happen. Sentence 2 actually did not find HeLa, but did find this raw 264.7 cell line. And so in this case, you can see that essentially the algorithm, looking at this particular sentence, actually missed one of the cell lines that was being used. So these are the kinds of mistakes that can happen with a text mining algorithm. And the rates are, as I said, the false negative rate is roughly 30%, while the false positive rate is about 5% overall for the particular algorithm. So now what we did is we asked across the entirety of about 2 million papers in the open access literature, and this was done last year in 2018. So you can see actually that the 2018 here is a partial year. We asked how many cell lines are there in all of the literature. So you can see however many number of cell lines there are. Certainly there are many fewer papers in the early years. Certainly there are more papers in the later years. These are the open access papers taken out of PubMed Central. And what we've done here is we've just looked at all of the instances of cell lines being caught by SCIScore. And then what we did is we had a list of the problematic cell lines. And we simply matched. We matched using several different criteria, but essentially they showed similar things. Of course, the loose criteria was, showed more cell lines that were possibly problematic, contaminated, or misidentified than the strict criteria. The strict was an exact match of the exact term. The loose criteria was a bit of more of a wild card match. So there's probably, the truth is somewhere here in between, and then we actually used a third criteria, which roughly was in the middle of these two, as that seemed to be the most reliable. But generally speaking, if you look here, this is the number of actual cell lines that were mentioned in any paper. And what we can find here is that there are, for example, in 2016, there are over 50,000 cell line mentions. And out of that, the percentage of cell lines that are problematic here are between about 5.5% or so and 10%. So the raw cell lines themselves have this kind of a distribution. And about 5 to 10% of them actually match one of these problematic cell lines that are listed by Icy Lag. Now, Icy Lag published a lot of papers. There's kind of a core paper that was published here in 2012. And so we tried to break the graph here, and we did our statistics on the two sections, because we were interested to find out whether the publication of the Icy Lag standard actually changed author behavior over time, over the entirety of the literature. And in fact, we do have a little bit of evidence. This slope is certainly different than this slope. So it does appear that, if we look in the early 2000s, generally speaking, there are some broad ups and downs here, probably because the sample size is relatively small. But definitely we do start to see, an increase in the incidence of these problematic cell lines being used. And while the average in more recent years is higher than in previous years, you can see that the trend is actually starting to move in the right direction. This will certainly be played out over the next few years, so we should be able to start seeing, hopefully, a faster acceleration of this slope. But this is generally what's happening. So it does appear that Icy Lag is having an effect. However, that effect is rather slow. We looked also across different journals. So there are some very good journals in here. The thing that makes the journal come up on this particular slide is this is the top 25 journals ordered by the number of cell lines that actually appear in that journal. So these numbers are actually quite large, and of course only those that are in the open access literature. So there are certainly some very good journals out there that we did not test because we could not get access to those. But you can see here in +1, and there are two bars here. So the first one is the cell line. So the cell line itself, number of cell lines in a +1 paper, or the percentage of cell lines, sorry, on the problematic list in a +1 paper here is about average for the entirety of the corpus that we looked at. It's about 8.5% or so. And you can see that varies a little bit here and there. But what this shows, the second bar, is that this is the number of papers, so the percentage of papers and the percentage of cell lines themselves. So the reason that the percentage of papers is higher is because most authors will use multiple cell lines per paper, and maybe one of those cell lines is actually on the problematic list, but then if one of the cell lines is actually on the problematic list, then the whole paper may need to be looked at again in terms of, the conclusions made by that paper. And so when you look at the percentage of papers here in the yellow bars, this is actually quite alarming. This is, the potential for between, 15 and 20% in most cases of the biomedical literature of papers that are actually using cell lines, where the authors are using at least one cell line on this problematic list, which means that we may have to reinterpret some of those results. And it's not extraordinarily clear which of those results at this point, which of those, nearly 1 in 5 papers will need to be reinterpreted, because cell lines on the problematic list, for example, if they're misidentified, they will be potentially used very effectively by one group of researchers, as long as the researchers know which cell line they're actually using. But if they don't know which cell line they're using, they think they're using one cell line and they're actually using a different cell line, then their conclusions are not correct for that particular disease. But I mean, the results are still the results. But if the cell line is a hepatocarcinoma versus a hepatocytoma, it may change what the results actually mean for medicine. Okay, so how do we actually make, this is a big question, look at the Icy Lag List before they publish their papers? So here is one example where an author had to look at a particular cell line. This is in a Cell Reports that was recently published. It's published in January, profiling human pancreatic beta cells. And what you can see when you look at the STAR methods section is this table. So the table says, what is the thing that I'm using? Where did I get it? And what is its identifier? And this identifier is kind of this nonsense string of letters and numbers. But when you have this nonsense string of letters and numbers, you can do some interesting things with it. This is the persistent unique identifier for this particular antibody or this particular cell line. So let me show you what you can do. If you go to Google Scholar with nothing but that number, you can actually start to see what are the other journals, what are the other authors that have actually published with this particular antibody. So in this case, in that particular paper, this particular human antibody, AC antibody, from Abcam was actually being used in 129 other publications. And you can see those here spread across all different, you can see this is from Wiley, this is from Nature, and this is another Elsevier journal. So how do authors actually get these RRIDs? Well, it turns out that authors are actually directed to rrid.site or scicrunch.org/resources, which really contains a good set of data from many, many different stock centers and data repositories, including, I would like to show you here, Icy Lag. So there's one consistent search, which is uniform across publishers. There's one set of instructions for authors, and there's lots and lots of data, all being represented in one portal. And of course, we do have a help desk. But you notice that the Cellosaurus data and the Icy Lag data are included. Other things that are included in the RRID portal: antibodies, plasmids, cell lines, including the IIDP cells, transgenic organisms, there are a lot of those. And of course, we have software projects, databases, and core facilities, and there are nearly 16,000 of those. So this is what an author might be faced with if they try to find and look up this RRID in the resource identification portal. So if they search for their cell line, Colo 720E, what they will be faced with is a record which says, here's how you cite this record. This is how you cite this cell line. But there will be a comment, and it will say, problematic cell line. This one's been contaminated. If they try to find that particular RRID on the Cellosaurus website, which reflects the RRID, and by the way, this is the source for RRIDs for cell lines, the same exact warning is here. And there are certainly PubMed IDs in both places showing where this was found to be contaminated and how it was found to be contaminated. So now the question is, how many papers with RRIDs actually have these problems? So when a set of authors is putting in these RRIDs, they've presumably been able to see that warning. And do they still use the cell lines? So here's an example of how we can actually find and how we do find all of the RRIDs that have been used in a particular study. So This is a study where we basically have looked and annotated by using a semi-automated method all of the RRIDs for all these various cell lines being used by this group of authors. And those annotations are made into our backend systems, and they become part of the record. And you can see that this particular cell line was actually used in 22 different papers. But all of this information is actually available to us, and it is, of course, available on each one of the pages on SciCrunch for each of the cell lines. And so when we actually looked at the overall number of papers that had one of these cell lines that were on the Icy Lag list, we found that about 5% of papers, where the authors used an RRID, were matched one of the problematic cell lines. But that is really compared to the text mining results, which showed a 16%. So 16% of papers versus 5% of papers, and presumably these authors already saw that warning and decided that the warning was not critical for their particular experiment. So the ends here are very, very large. This is 150,000 papers, and this is 634 papers, making the difference quite significant. We've certainly been asked whether there are confounding factors here for the RRID project, and there certainly could be. But the number of papers really suggests that if this is, if all of these authors are better authors than these authors, then there's certainly a whole lot of really good authors here. So we think that this sample size is very, very highly significant. And certainly the difference is over 60%. Okay. So let's take a look at how many papers in total there are already using the RRIDs. And this is a graph I like to show, and it really shows where the project started with 100 papers at the end of 2014. And in 2018, we're about 12,000 papers. And importantly, endocrinology was one of the journals that joined in late 2016. And we'll take a look at, the RRIDs actually used in endocrinology. So endocrinology started by enforcing RRIDs for antibodies. So just about, and this, you'll notice this is a log scale, just about all the RRIDs in the journal Endocrinology are being used for antibodies. However, recently the journal has actually shifted the policy to also start asking for transgenic organisms and cell lines. And we are starting to now see many more tools, such as software tools, cell lines, such as you have seen, and some of the organisms. So this will continue to shift. The overall project statistics appear that about 76% of the RRIDs are actually being used for antibodies. But the cell lines in 2018, this was tested in 2018, so things have changed. But generally speaking, cell lines at that point were about 3%. We're expecting that to be probably around 5% currently. But this is essentially what we're starting to see. Certainly, cell line portion of this is growing, and it certainly will grow within endocrinology publications. Okay, so what can RRIDs do for me? If you have a research resource, like you run a stock center, or you run a core facility or one of these, you can actually include RRIDs for your important resources. And then the tracking will become much easier in terms of how is this supposed to be cited. So here are some really nice examples. Of course, you saw the XPASI, the Cellosaurus data already, and you can see the RRID is right there for how to cite this particular line. This is another one that's contaminated. So presumably you would not want to cite this one. But the MMRC has this on each one of their mice, and you can actually copy that by just clicking this button. If you run a stock center or a repository, you can get your RRIDs, but even if you run something like a core facility or a project, you can just make this very explicit. So this is the acknowledgement language to go into your publications. If you are using any of the resources from HERN, you're asked to acknowledge this particular RRID, which is the part of the tool section of the RRID initiatives, and that helps the people who are trying to cite you. It clarifies how you would like to be cited, and then that information again becomes part of this record. So where are we going to go from here? So we have asked the question, will journal editors and authors be willing to go the extra mile to actually do this checking and adding of RIDs for cell line authenticity? We believe that we certainly have a compelling reason for them to do so, because it is an easy way to check the Icy Lag List as opposed to downloading it from the Icy Lag website. So we think that it will drive adoption, but we will certainly be looking for that in the next couple of years. And then the second portion is we have run the SCIScore algorithm on all of the open access literature, but we have also recently made the SCIScore algorithm available for authors who can sign up using their ORCID account, and they can run their own verification method on their papers. So we would like to see if SCIScore will actually help authors identify these cell lines earlier. Okay, so this brings me towards the end of my discussion. So I always, instead of thanking all of the wonderful people that helped us do this work, who do definitely need to be acknowledged, but one of the things that I always like to do is to have everyone walk away with an action item that I would like to ask all of the people listening will be able to do. Love to have everybody put RRIDs into their next paper to check and verify whether your cell lines are authentic and to make sure that your antibodies are well identified. If you have a particular project, bioinformatics project or anything else, or a core facility, put an RRID in an acknowledgement text for your particular center. This will help improve the number of RRIDs in the literature, but it will also improve how and elucidate to authors who might be working with your resource how you would like to be cited. And of course, when you review papers and you review grants, we ask that you pay more attention to the methods. And if you have a journal, I would love to talk to you. My email is down here. And I would like to thank you all for listening and open it up for questions, if there are any. All right, well, if there are no questions, I am going to sign off and invite you to our next webinar for the Signaling Pathways Project, Putting the R in Fair Data by Dr. Scott Aschner. And I thank you very much for your attention.